Gordana, do you see my screen?
Yes, everything is good.
Okay, good, perfect. And good morning, everybody. Thank you for participating today. My name is Sevil, and I'm a postdoc at DTU Sustain. As Gordana already said, I'm doing research on the fate and effects of bioplastics in the environment and in the waste management system. And for the presentation today, I will focus on biodegradable microplastics and the extraction from compost. And this method was developed during my PhD at DTU Sustain and with collaborators from CNR ISP in Venice. The method has not yet been published, so the recording, I guess, will be stopped or paused for this part of the presentation. Right, Gordana?
I can make a break while when you tell me.
Okay.
Okay.
Yes. So first of all, I would like to start introducing biodegradable plastics. And biodegradable plastics are a subgroup of bioplastics, which can be seen here in the figure. Let me just do like this. Yeah. So we have bioplastics, which are all these things, and then biodegradable plastics in here. And they can be both bio-based or fossil-based, but in both cases, they are designed to be biodegradable. So this sets them aside from conventional plastics that we have found here that are always non-biodegradable. However, these biodegradable plastics, they need specific conditions to biodegrade. And if those conditions are not met, the biodegradation may be incomplete, and the plastic can therefore fragment into microplastics without obtaining complete degradation. So this also means that we actually, for biodegradable plastics, can have more microplastics produced from these types of plastics compared to conventional plastics. So today, less than 1% of the plastic production consists of biodegradable plastics, but this is expected to increase in the future. So it's very important to kind of understand anything about these biodegradable microplastics, which have not been that much investigated compared to conventional microplastics. So compost is a common end-of-life environment for biodegradable plastics. Many biodegradable plastics are marketed and labeled as compostable, meaning that they would be able to biodegrade under composting conditions. But to fully assess if biodegradable plastics degrade completely in compost, we need to do microplastic analysis to see if there are any microplastics left in this compost or if biodegradable plastics are completely degraded. This can, however, be difficult. As you see here, we have a photo of a real-life compost, and it's a very complex matrix, making it very difficult to kind of investigate microplastics if the compost sample is not pre-treated before. So before kind of being able to investigate if there are any microplastics in a compost, we would need to introduce biodegradable plastics to this compost. And in my PhD, I also did a degradation test of different biodegradable plastic products. These shown here, different types of biodegradable plastics and different products, so some bags and some flower pots and so on. So the question is it completely degraded, or do we end up with microplastics in the end? For this presentation, I will focus on this PHA-based bag, as we did the oleo-extraction method for the compost that had been exposed to this bag. So I will quickly just go through the degradation studies. where we tested biodegradable plastics using ISO standard 2200, which simulates industrial composting conditions. So according to the standard, after the test, particles below 2 millimeters are considered as degraded, and particles above 2 millimeters are considered non-degraded. And looking at this photo, we have the plastic before the test, and then after the composting test that had a period of 90 days, we then sieved the compost above 2 millimeters, and then we still had these microplastics that were then not degraded. If you want to go into the results of all the plastics, you're welcome to look up the article that we have published here with this QR code and find the results. But then what is interesting today is now the fraction below 2 millimeters, the compost here, where according to the test, plastics in this fraction would be fully degraded. But actually, if you can see, I can see a little piece here of plastic. So this compost fraction below 2 millimeters actually also consists of plastics, but not macroplastic, but microplastic. So we then continued with this fraction below 2 millimeters. And before doing any treatment to this fraction, we tried to look in the microscope, and we could identify some microplastics, but it's very difficult to identify plastics with this complex matrix being in the background. Some particles can be hidden under the compost matrix. So extraction is really important to get now a clean background where it's easy to identify the plastic particles that you're interested in identifying. So some challenges exists with the biodegradable microplastics, because the existing methods for extraction are often only suitable for conventional plastics because they involve harsh chemicals and are conducted under elevated temperatures. And these conditions can be too harsh for biodegradable microplastics because they can end up degrading during the processes of the extraction, meaning that we actually destroy the particles that we're trying to identify. So it's therefore, when you're looking for biodegradable microplastics, really important to kind of think of a method that is mild, where you don't end up destroying or harming the particles that you want to identify.
The twinning part of our project, where actually we do capacity building and staff exchange between institutions to showcase how actually we are proceeding with our project and the results of InPlasTwin. We have time for some questions which might arise, so please use chat if you have some questions addressed to Sevil's presentation. Okay, so far I don't see questions, so we can leave them then for the Q&A section. And if you all agree, we can continue with the presentation on the 4th matrices that we will cover today. It's from Biota, and the presentation will be given by Dr. Eleni Christoforou. Eleni, could you please share your presentation with us?
Hello, everyone. Can you hear me and see my screen well?
We can hear you, and we can see your presentation. Thank you.
Perfect. Thank you. Great talk so far. So I hope you're not too tired to have the last talk for the day. And we're going to talk about tracking microplastics in Biota, hands-on sampling and extraction technique. I'm a marine biologist at the Cyprus Marine and Maritime Institute, and I work on the PUREEF-Y project. So just a very quick outline. We're going to talk about the species selection, sampling, dissection, and extraction. And as the previous talk, I'm going to leave the analysis part for our next workshop that will be all about the analysis of microplastics. So let's get started. If you go on Science Direct, as you see on the right-hand side of my screen, maybe I'll put the cursor as well. So just writing microplastics in Biota without talking about both terrestrial and marine, you have 7,385 studies. So it is a hot topic, and you don't need me to convince you. If you're here, you know that microplastics are a very important topic. When we refine the search to marine biota, we go about 1,000 searches down, but still also on marine biota, it's super hot and super important. So in this case, we're going to talk about how to find and identify which species you would like to investigate and what are these conditions and considerations. So first of all, it's important to look into indicator species, and that would help by looking into the feeding mechanism of the organism, what you want to study, their habitat and utilization, if they're sessile or they're mobile species, what's the literature out there, and if there's proof that microplastics are ingested through laboratory work. That makes it much easier to refine your methodology. Then, of course, the geographical distribution. If I'm in Cyprus, it will be of no use to have, to investigate a species that's far, far away from here and where my study area is. The abundance of the species. If you could investigate a species that maybe it's alien or invasive and it's causing a problem to the ecosystem, it would be much better than tracking and collecting species that are endangered and you don't want to harm them. Of course, ethical conditions should be considered, and also the collection effort. If I need to take out a huge trawler boat, or if I can collect them manually by hand. Of course, the relevance to the ecosystem services they provide, and if they're consumed by humans, are they not consumed by humans? What's the main question of your study? And of course, the bioaccumulation with which that would come from literature information. And a very handful and useful tool is the Harmonized Protocol for Monitoring Microplastics in Biota by Bessa, Philip, and al. And something they mentioned is that bivalves, in particular, the mussel of Mytilus genus, does fulfill most of these requirements and the criteria mentioned, as they are abundant in intertidal and coastal locations. So they are filter feeders, indicators. There's a lot of literature about them. They can be found worldwide, around the world, and they can be very easily collected. And by being sessile means that you do investigate that specific location and you don't get information about, different, if they're migratory species, you would get information about different locations. So it's hard to distinguish where the pollution factor is happening. So with this in mind, and just not to be based by literature, but we do see the problem ourselves. So this problem can be seen here. If you see the mussel, you see a fiber that's been trapped within the mussel, and that's a photo taken in Cyprus. On the top right, you see a lot of microplastics floating at the coast of Scotland. And on the bottom right, you see mussels that were formed colonies on the platform, and you see all the nets and the nylon threads that are all around the community. This is maybe a good time to say that in PUREEF-Y, we will be investigating microplastics in Biota, but as we're still in the first year of the project, we haven't gone through it yet. but we will be using the method that I will present to you, which I refined during my PhD, and hence the photos from Scotland. And here's the information. So the next most important thing about investigating microplastics in biota is to know the biology of the organism and what you're interested in finding. So we're going to have a really quick run through on the biology of mussels. So mussels are filter eaters, and the particles that enter through the inhalant bulb, like here, which is the rough area of the mussel, which also restricts the size of the particles, which is a very important topic to consider. So particles coming in through the inhalant valve, they would go into the ctenidium, which is the gills. They will pass through the ctenidium, and then they will run into the food groove. The food groove is like a little channel, let's say, that they would pass all the way to the labial pulp that helps with the sorting. So at the labial pulp, particles that are not food items will be rejected, but that's an interesting topic again, because some microplastics are not rejected into the pseudofeces, but they do reach the stomach and the digestive gland. Through there, anything that's digested and remains in the organism, while the rest would leave the animal through the exhalant valve. Going back to the rough surface, that would determine the size of phytoplankton, which is the food item that they usually eat, but also of microplastic. The one tricky thing, though, is that fibers do have a little thin area. So even if the mussel cannot take up more than maybe 100 microns, some microfibers would go through their narrow area, so you would find bigger fibers in the mussels. Then going to the collection. So when I developed this methodology, it was developed for laboratory experiment. And at the end of this presentation, I'm going to go through some notes and considerations for when you're collecting the organism from the field. So first of all, we collect the mussels one by one, and we close the valve by wrapping them in a rubber band. So that's a very, very important step, because you close the organism and whatever is in there remains in there. So that eliminates cross-contamination and anything that would go around. There are other studies that use aluminum foil, but sometimes aluminum foil cannot be wrapped strongly enough and the mussel could open again. So it could introduce contamination, something that the mussel didn't have inside. So after that, you measure the shell size of the individuals, basically. In this image, you see measuring the height, you would measure the width and also the length of the organism. And here it's important to mention that some studies think about and consider about weighting the mussels, but I don't agree, let's say, that much, because in mussels and in bivalves, water is retained inside. So by weighting it, you don't know if the shell was full or empty. So in this case, it would be best to measure the shell size, which is analogous most times with the organism. And then that you place the individual in pre-labeled freezer bags. At this point, you don't mind if they're plastic, because you have already wrapped the mussel. So anything around, it's not contaminating. And you, of course, place it in a cool container, and then you freeze the organism at -20 until dissection. Freezing the organism immediately after collection is the most ethical way, let's say, to kill the organism, because they're not sensitive to temperature, so they would smoothly die instead of allowing them to die by themselves. And also, you keep the organism fresh for easier dissection. Then going to the dissection, you first defrost the mussel at room temperature. It takes about 30 minutes, and you remove the plastic band. You open the shell, but you need to open it slightly, not like open it all the way. And like you need to open it enough to be able to put the scalpel inside the shell to cut off the muscle of the shell. Looking at this image, the muscle is this white part here. If you open it, I would say violently, if you open the whole shell, then you will destroy the tissue inside, and it will be very hard to dissect the digestive gland and stomach. So you remove the shell entirely, and you end up with something like this in the bottom left image. And you put it under running water. In this case, we're only interested in what's been digested. So anything around, again, we don't mind. And you place it in a Petri dish and dissect under a stereoscope to remove the stomach and digestive gland. This is one of the best dissections that I was able to do. The stomach is basically this white part that goes inside the digestive gland, and the greenish brown stuff around is the digestive gland. It's almost impossible in my experience to get only the stomach, hence we have this part. And you see here the stomach and the digestive gland. And based on the anatomy we saw, here are the labial palps, and the valves will be up here. Again, this was performed based on an experiment, and maybe it's important to mention that we added and inserted our own microfibers that were made out of nylon. So we knew exactly what we were targeting. So we're not very scared of cross-contamination because it wasn't something that could be in the atmosphere. And that's why I'm going to go into more details on being more careful with cross-contamination when you're dealing with organisms from the field. Because again, there you have like a variety of fibers and plastics, microplastics and microfibers. So after you have the digestive gland, you prepare a trypsin enzymatic dissection. It would be 0.3125%. It's a tiny amount of enzyme in deionized water. And you place it in a glass beaker where you would place the digestive gland and stomach. You place it in a magnetic stirrer with small magnet at 45 degrees, medium speed for 30 minutes. It was, most of the cases, enough time, but if you do still see pieces of the organelles, then of the organs, then you could keep for some more time at the same temperature and speed. Then you pour it into a centrifuge tube, as you can see on the right, and use a squeeze bottle to ensure the full transfer. And then you centrifuge at 305,000 rads per minute at 15 degrees for 15 minutes. And the result is what you see at the right. You would pipette out all this liquid, the supernatant, leaving about 1 centimeter above the pellets that are created. The reason you want to leave some above is that you don't want to disturb it, and otherwise you would need to analyze more. After, you would homogenize the remaining pellets and the little supernatant that you left and place a drop at a time. It usually takes about 3 in total on a glass slide and cover with the cover slip. And you would view under the microscope. In this case, I could view it under a 10 times 10 magnification. And here's what you see. You do see the fibers. These fibers, they're about 30 micrometers in length. So you could see like the small ones. And then you use a zigzag pattern as usually in microscopy to cover the entire slide and quantify the microfibers. And of course, you repeat for the rest of the sample. Hence, I said earlier, you wouldn't want to have more because it will be more time consuming. And just a note, because I think it would be a question, that discarded supernatant, the liquid that you saw on top, was tested not to contain significant number of microfibers between the samples, hence it could be discarded. So some benefits and limitations of the enzymatic digestion, because we saw with previous presentations that, of course, any method has its limitation and there are different methods. So the benefits are that there is minimal to no damage of most polymers preserving their color, size, and shape. And the reaction is occurring under mild conditions. So it's not harmful to work around it, and it's usually like 30 to 60 degrees. So no risk of burning, or pH neutral conditions, and it has a low environmental impact. On the other hand, though, it's not appropriate for large tissue sizes. So I cannot put a whole fish in a beaker and try to digest it. It will never happen. And it's an expensive method. It's more expensive than acid digestion or base digestion, because the enzymes are expensive. And it's time consuming, require multiple enzymatic steps, as we saw with the duration of time. And it targets specific organic materials, so protein, cellulose, and lipids. Now coming to the considerations for field collected samples. It's very, very important to reduce the airborne cross-contamination. So you need clean air facilities in the laboratory, consistency in the PPE used in the laboratory, so that you could analyze any contaminants or microplastics that you usually see that could be blamed on the equipment used in the laboratory. The use of glass containers washed with filter water and covered in aluminum foil is very important, because again, you don't want any airborne microplastic to be interfering with your research. And monitoring is also very important, considering blanks and controls. The selection of extraction process is also important. As I said, it depends really what you want to extract, the size of the particle of the organic matter, and what tissue. For example, potassium hydroxide is not appropriate for fat tissue, but it is appropriate for other digestions. Time consideration, sample size, and the anticipated microplastic type. So again, some acids, when I was investigating, they would dissolve or destroy nylon thread. So that would not be good for the experiment that I was running. And in terms of the analysis, the preparation of the samples for micro-FTIR analysis is also an important factor. You would need to get the appropriate filters, usually metal filters, for the specific machine that you have. Further digestion may need to be required in case it's shadowing or creating a layer on top of the microplastics, and the analysis of any commonly used material sources of contamination, as we discussed. For example, if you're using a bucket, it would be good to test the type of plastic bucket that you're using, if you cannot use a metal one, of course, to know that if you see that in your samples, you would consider cross-contamination or not. So if you have any questions, please feel free to ask me. I'll stop sharing so I can see you all. Stop sharing.
Thank you. Thank you, Eleni. It was very interesting, with very interesting photos. I have to say that, yeah, congratulations are just coming from the participants. What to say after these 4 amazing presentations, that we have dealt into really a challenging topic of how to sample and analyze complex matrices, and to highlight the necessity of having the standardized methods for each of these matrices, which is now a research topic among all institutions, I'm sure, that are dealing with microplastic and nanoplastic research. After Eleni's presentation, I would like firstly to ask, are there any questions for Eleni? And then we can move to next from the participants. So maybe while we are waiting, we can start our Q&A section from Eleni, maybe, because or you need some break.
No, I'm fine. I'm done.
What might arise as a question after your presentation is when you are using alkaline or acidic digestion for biota, how actually do you determine the optimal balance between complete digestion of tissue, particularly calcified or tough matrices, and minimizing actually the damage or loss of the target microplastic polymers?
So first of all, I would base everything on literature and what has been done by other scientists. Of course, we don't want to discover the wheel again. So first of all, yeah, determining on that. And then I would try first with the minimum amount of acid or alkaline solution, or even as I presented, I would even go with the least harmful, which is enzymatic digestion, and then see if that is not working, try to increase the strength, let's say, of the solution that we were preparing. And of course, also try to increase maybe the time, the temperature, so play around those factors before going to more acid solutions, because we know that acid will destroy the microplastics further. Another thing that I would consider is, because for PUREE-Y project also, we do collect also sediment and water samples. So from the areas that we collect the biota, I would consider what's in the water and what's in the sediment to have an idea on what microplastics I should be targeting. And exactly. So if I look into literature and say that the, I don't know, 80% acid solution destroys all the nylon, you know, and I don't see any nylon in my water samples, then I'm safe to use it, you know. But if I see nylon, I'm just giving a random example, but if I see nylon, then I would consider other methodologies.
Thank you so much for the answer. And what also comes to my mind while looking at the photos and what actually you passed through your PhD, we can see that the samples actually from biota are highly susceptible to some kind of external contaminations. So do you maybe have some tips for minimizing this kind of contamination during the initial sampling and dissecting the small microorganisms?
Mm-hmm. So it again depends on the type of organism and what you're targeting. In the case of bivalves, just shutting the valve as soon as you collect it, it's the easiest way because the valve will be impermeable. So as soon as you literally wrap the band around mussels or any oysters or any other bivalves, you do retain everything in there. So that's not an issue. And then when you go to the laboratory, working under standardized conditions like fume hoods, eliminating traffic around you, turning off any fans or closing windows, you know, these normal microplastic practices would be very helpful. Also having a control near your work. Now going to other organisms like fish, for example, if you want to get, like, try and see microplastics from the gills, then it'll be very hard to eliminate anything that passed by, you know. So it would need to be immediately sealed in a bag. I don't think there would be metal bags. So you would need to consider the type of bag that you're using and be consistent. So again, if you see that type of contamination in your results, you would know where it came from. But again, if you're looking in the inside of the organism, so in the stomach of fish, then you wouldn't be very concerned on the outside contamination because nothing will be eaten by the fish by the time you take it to the lab. So it's really case-by-case scenario.
Thank you for these tips. I'm sure that they should be useful to the ones who are dealing with biota, and hope not to push you much with questions, but maybe one question more. As you said, later on we will announce our next workshop, which will most be dealing with analytical techniques. But for example, when you are doing sampling extraction protocols, do you account for potential chemical like alteration or degradation on microplastic due to digestion protocols? And how then this can affect the identification by techniques like FTIR or Raman techniques?
So this would be more considered, I guess it's what I said earlier, that you first have to start with the less hard and harsh conditions before you go to the chemicals that do destroy the plastics. So I think that's the only way to just know what to expect and be able to work around the plastics that you expect to see. And that would save you some trouble. But there is no real way of knowing exactly on what you're going to find and how to refine your methods. It's, yeah, you need to try and test.
That's something that is always exciting with the researchers when you do something and when you are finding out how to actually tackle and go on with something. And of course, if you find a solution, then to share it and spread the word among the research community. Thank you.
And may I add, sorry, just a small thing. Maybe it would be helpful to, especially we're developing new techniques, to get more samples. So if you're working with the muscle of fish, you have plenty of it. You know, it's not one digestive gland and stomach of one muscle, you know, so you can run small tests and small experiments to see what's the best way.
Certainly.
Yeah.
Thank you.
Thank you.
Thank you, Eleni. Maybe some more questions to Eleni? Nothing arises now. I would also like to give floor to our other speakers to discuss other matrices that we had opportunity to hear about. So if you agree, I would start with Nu. So may I ask you to elaborate more on the most critical contamination control points you enforce during the sample preparation phase for soil, which is often done outside of this clean room as a clean room environment?
So you mean what is the most sort of contamination, or what is
yes, because I know that some laboratories maybe doesn't have these special clean rooms. So maybe can you comment on this?
Yes, so like we like wearing 100% cotton lab coat could prevent at least contamination from the analyst to the sample. And then for airborne microplastic, then it should be good to also cover all the glassware and all the thing that going on with aluminum foil. But there is also one study saying that aluminum foil can be also a source of plastic contamination. So that's why we normally also bake. So put it in the oven and burn before we use that aluminum foil to cover all the what is going on in the lab. And yes, and of course we always have to use blank because it's really difficult to have like no plastic contamination at all. But with a sufficient amount blank, we can know like what is the baseline in the lab and then subtract it in what we found in the samples.
Thank you, Nufa. I saw that among the participants, we have one colleague that I used to work with. It's Ana Mendes, and I remember that she told me that also wearing cotton lab coats in the colors like pink or blue color can also be very effective for further analysis of microplastic in the samples.
Before
yeah. So you can easily see where it comes from. Thank you, Nu. And maybe can we address something about the standardization and say something like what is the biggest methodological gap that currently prevents the standardization quantification of microplastics in soils across different labs? Do you have some knowledge on this?
What is the biggest challenge in the
Methodological gap? Yeah, yes.
Yes, so for soil, it's definitely soil organic matter, because like for most of for soil there are two things: inorganic particle is sand and clay, and then the organic matter. For those inorganic particle, like heavy soil particle, we should be able to remove most of them during the density separation. But then for organic matter, because they have quite similar chemical composition and also density with the plastic, so it's really, really difficult to remove the soil organic matter. And then if there are still a lot of soil organic matter, they can interfere with our subsequent analytical techniques. For example, for Raman, it will have like fluorescent background that will hinder all the signal of plastic. For FTIR, it can create a false positive result, especially for polyethylene. And similar with pyrolysis TCMS, where soil organic matter also can give false signal of polyethylene, polystyrene. So we have to really be able to remove these like really stubborn soil organic matter. But at the same time, if we spend too much time and effort trying to remove those, we also might damage the plastic as well. Or we like it take more time. The more time it takes, the more contamination we can bring to the lab. So it's like finding the balance of how to treat this organic matter.
Thank you, Nufa. After your answers, I may conclude that actually it's you really need to have patience and a lot of time to deal with all these matrices. And now I would switch to the sediments if you agree, if there are no more questions to Nu. At the moment, I don't see. Fred, could you maybe elaborate more on what is actually the critical difference in sample handling, like storage and transport for marine sediments, that must be strictly followed to prevent contamination or degradation before they reach the lab separation stage? Because we have seen that you have your boats while taking the samples, which I had the opportunity to see and to visit, and I must say they are amazing, the equipment that you have. So can you maybe just address this, although you told and point in some things about this during your presentation, but I think it's important to tackle once again.
Focus on a multicore that is mostly used on...
Can you maybe put your mic louder?
The microphone.
Yeah, perfect.
You hear me now?
Yes.
Yeah, for the multicore that is widely used by us, the pipes are sealed on board the ship, and nowadays we don't cut it into slices on board the ship. It's transported to land upright, not frozen, but chilled. If you froze the pipe, it would disturb the strata within the pipe. And also it should be transported upright. And when it comes to land, it will be subjected to an X-ray to see if the strata is disturbed. If the strata is disturbed, then the whole sample is wasted. We don't use it. If the X-ray say, oh, this is perfect, still perfect, then we can take it into a lab or big room that has been cleaned, and the people that is doing the slicing is wearing cotton equipment, and they set up air blanks in order to see that the atmosphere is okay during the slicing procedure. The samples are transferred to clean glasses covered with aluminum foil, and the lid is put on and transported, and that is happening in Trondheim. It's in the middle of Norway, and it's sent to Bergen and put on a cool place, not freezed, but in a cool place. So we have little or no growing of moss or algae, because the method we are using does not cope for algae growth. And then it's brought into the sediment separation lab and handled there, with also all clothes in cotton, lab coats. Of course, we use nitrile gloves, and we analyze for nitrile, but we don't report nitrile because it could well be from our own equipment. Likewise, we don't report PTFE because we use PTFE gaskets and PTFE filters for the analysis. We don't report the silicone that is part of the hoses we are using, and the prop. So, yeah, that sums it up, so to speak. I think your first question.
Yes, actually, it's you already mostly gave the answer to my second question, which was is actually to highlight that marine sediments contain often a high fractions of slate and clay.
Yeah.
So your chosen technique at EMR actually by using these filters can effectively distinguish and separate microplastic in the specific size range from these dense non-plastic inorganic particles, right?
Yeah, if you're not careful when you transfer the suspension onto the Biota, the separation rack, then you might have a lot of silt and clay that gets into the compartment that should only contain the light or low density particles. And that has happened. And the solution to that problem is to do another density separation. If you have foaming on the suspension, that could well contain minerals, and that is not unavoidable. So from time to time, we see many or some silicate, probably silicate minerals in the light fraction. However, we are using the PTFE filters in the Spero instrument, and that means that we have a larger area that we can measure than on the FTIR. And if there are a lot of particles, especially coal particles, I can't do anything with them with the present procedure. We distribute the particles onto several filters. So we don't take every particle on one filter, but we distribute them on 2, 3, or 4 filters.
And that's how you actually overcome this.
Yes, yeah.
This problem.
So I seldom see that it's perfectly clean and only plastic particles. There will be some mineral, some organic, some plant residue, or animal residue on the filters.
On the filters.
Yeah.
Thank you, Fred. From my side, I don't have any more questions that might arise. I think everything was explained in very much detail. I'm looking in the chat. I don't see more questions to Fred. So after covering soil, sediment, and biota, I would like just to return back to the compost and actually this oleo-extraction, which is presented by Sevil as a promising for biodegradable microplastic. It's actually in the focus of InPlasTwin project. Sevil does this process actually show high selectivity toward a certain polymer types, and how is actually this method validated to ensure high recovery of a broad range of biodegradable microplastic, which are present in compost, as you already said during your presentation?
Yeah, so for the compost, we only test this one type of biodegradable plastic because as opposed to the other presenters, they take an environmental sample, but we have a compost free of any plastic and add these large amounts of large microplastics and then try to find these exact plastics. So we know what we are looking for, and we only tested this one biodegradable plastic. And then for the recovery test, we used the same biodegradable plastic, but then also another plastic, which was a conventional plastic. And for the recovery, we saw that both plastics got a high recovery. The biodegradable plastic had a recovery of 88%, and the conventional plastic had a recovery of 83%, so both very high. We have not tested with other biodegradable plastics, so we don't know for that, but that can be done and will be done. And
will be done in future investigations.
In the future. Yeah.
And as the same as soil, and the compost is also very rich in organic matter, like No mentioned. So how actually this oleo-extraction process managed to co-extraction of natural fats, waxes, or oils that comes from compost that can be present, and what actually are there any necessary subsequent steps needed to ensure a clean final isolate which will be ready for further spectroscopic analysis?
So before introducing the oil, we have the sample pre-treatment where we use the hydrogen peroxide. There, of course, you can play around with how much you need to clean your sample. We tried 3 different concentrations, or 1 without any pre-treatment, and then 2 different concentrations of hydrogen peroxide. And yeah, so adding these 5 milliliters to 100 milliliters of water provided a clean sample in the end. Then we also tested with 10 milliliter hydrogen peroxide. This gave an even cleaner sample, but what we saw was that the biodegradable plastics that we were looking for were degraded. So it's a balance between how much you need to add to get a clean sample, clean enough to analyze, but not so clean that you also destroy what you're looking for. But yeah, for compost, this was enough. Now testing it on soil, we still have some issues. We get a dirty sample. We need to try to see if we can add more hydrogen peroxide and less than the 10. But then also maybe introduce some density separation. We'll see.
So a lot of work and a lot of testing is actually needed for dealing with microplastic particles and their analysis. If there are no more questions, I would now share once again my presentation, just to return and to maybe make this last minutes of our workshop to make it maybe more proactive. So as a continuation of the workshop that we had today, we would use this opportunity to announce our second workshop, which will be organized in synergies with PUREEF-Y project. So the coordination of whole workshop will be done by colleagues from PUREE-Y, and it will be connected with advanced spectroscopic approaches for microplastic detection and characterization. So today we dealt with sampling and extraction, so preparation of samples, and our second workshop will actually deal with how to analyze these samples. All the informations will be available through both PUREE-Y and InPlasTwin project websites, and as well as our social media channels. That's why I have put the QR codes of InPlasTwin project, which you can follow. And at the beginning, you had the opportunity to see what social media networks you can follow from PUREE-Y project. So just to keep you updated, remember and mark in your calendars that the 19th of January 2026 is envisaged for the workshop 2. It will also last 3 hours. We will have 4 speakers that will actually deal with different spectroscopic methods for microplastic detection and characterization. But not to finish with this workshop, because we have 2 more years ahead. We would like to ask all participants to write us in the chat answers to these 2 questions, so we can prepare the workshops in accordance to your main interests. The first question is, what is the single most valuable topic related to microplastic that you would like to see covered in our next workshops? And in order to prepare our speakers for the future workshops, we would like to know what is the biggest challenge that you face right now in the methodology or execution of your microplastic research, so we can try also to tackle these challenges for our future workshops. I will leave this now open and give you 5 minutes to write your answers in the chat, which I will gather further and analyze them. Maybe I will just exit the full screen just to see, so I can follow the chat like this. Do you see the questions?
Yes, we see the questions.
Okay, thank you. Thank you. I will be patient. Few minutes more just to get some feedback from the participants. I don't see any answers so far. I hope the chat option is working to everybody, maybe. So I assume that we will be more okay. We have a topic, the effectiveness of different methodologies and how to choose the best one on your samples. So, Leda, thank you for your comment. As you are part of Cyprus and Marine Institute, I'm sure that together with Kati and Gotelin, we can figure out on the next topic workshop on this, as you suggested. Selection of appropriate methods for digestion of tissues and of filters for AR microscopy. Okay, Vlatka, thank you for your suggestion. We will take this into account, especially for this second workshop to tackle also this question. You can leave the answers in the chat until we finalize this workshop. What I would like also is to call all researchers that are participating in similar projects and to highlight that both of our projects are open to synergies. And if you see any complementarity with our work, please feel free to write either me on my email you can find on the project InPlasTwin website, or to Kati Christoforou, who is coordinating, actually managing the PUREEF-Y project, and we will be happy to establish new synergies and new potential workshops together with your project. And at the end, I would like to thank you all for participation. But before we end, once again, I have to give my sincere thank to all the speakers who gave a big contribution to this workshop. Also to the organizers from PUREEF-Y and from InPlasTwin. I want to say that beside that this webinar will be uploaded to our YouTube channel. You can also contact all our speakers in case that you have some specific questions to the methodologies they have presented. And also all contacts can be found at our project web sites. Thank you to Dr. Janja Widmar as project coordinator of InPlasTwin project, and to Gotelin Piaton as a project coordinator of PUREEF-Y project that gave presentation of the projects and introduction to what actually we do at InPlasTwin and PUREEF-Y. Once again, thank you for all for your attention, participation, and looking forward to see you on our next workshop, which will be on 19th of January 2026. Till then, I wish you happy weekend, happy rest of the day.
Thank you.
Thank you. Bye.
Thank you, Gordana.
Thank you all.
And
Thank you and goodbye.
Goodbye.
Goodbye and thank you all.